Correct. Hello, everyone. Good afternoon, and it's good morning for me. So today I would like to present a talk on my PhD research that was about formulation and development of nanomicelles for breast cancer. And specifically, I did this for metastatic breast cancer and triple negative breast cancer because these are some of the most hard to treat and breast cancer with a high rate of remission and low survival rate. So in today's, I'm not getting the next slide. Yes, so yeah, today I'll be discussing I'll just give a brief introduction about breast cancer and the approach that we used here for my research on nanomicelles, specifically targeted nanomicelles for breast cancer, and the method in which the nanomicelles were prepared. And then I'll discuss the results in brief and then finally the conclusion. So giving a brief introduction about breast cancer. And so in 2017, there were more than 250,000 cases of female breast cancer and more than 40,000 females died from breast cancer. And a major impediment to breast cancer therapy is the development of multi-drug resistance. And this causes a low survival rate and the current treatment become ineffective. So also to overcome breast cancer, there are many therapies. And one of them is to make a nanoparticle or a nanomicelle that can cause enhanced permeation and retention effect. This means that the therapeutic agent lies close to the tumor tissue, and then it continuously leaches its contents into the tumor and microenvironment, which causes the enhanced retention and permeation effect, which is mostly seen in nanoparticles therapy for cancer. And a late stage breast cancer can develop into metastatic breast cancer when the tumor grows away from the breast tissue and when it touches the chest wall. This tumor then can metastasize to the bones and other organs, causing metastatic breast cancer. So in the past, there have been many formulations and many medicines that are developed for breast cancer, mostly involving the drug paclitaxel. Paclitaxel is one of the most commonly used cancer drug. And earlier, the first generation formulation of this was Taxol, and this was a oily solution of paclitaxel in chemophore. So this formulation made it difficult for the paclitaxel to reach the cancer tissues. To overcome this, a nanoparticle formulation of Abraxane was developed. So here paclitaxel was conjugated with serum albumin, and this nanoparticles was a better therapy than Taxol formulation. And recently, another formulation of paclitaxel is developed, which is Synveloc, which are polymeric micelles of paclitaxel. So here the drug to polymer ratio is higher as compared to Abraxane. So here in my research, our approach was to develop nanomicelle, which have a higher drug to polymer ratio as compared to Synveloc. And then these nanomicelles were targeted. And because of targeting, the nanomicelles can specifically attack to the cancer cells and spare the healthy ones. And there are some differences on the receptor that are expressed on the cancer cells as compared to a healthy cell. And if we harness that information and use it on our nanomicelles or nanoparticles, it can be a useful therapy for cancer. And then in my research, I also used a mixture of two drugs, which is ritonavir and paclitaxel. So ritonavir is a P-glycoprotein inhibitor, which decreases the efflux of drugs from the intracellular space of cancer cells. So this means that a paclitaxel drug will be concentrated in the breast cancer cell and thus cause the breast cancer cell to die. And here in this schematic, as you can see, when the cancer spreads, it spreads to other tissues, and then the blood vessels develop to supply the cancer tissue with blood. So here the reason why we use nanomicelles was because they protect the drug from degradation, and of course they increase the circulation half-life of the drug, and that can lower the dose in the nanomicellar formulations. Also, they have higher enhancement permeability permeability retention effect as compared to nanoparticles, and they can target the drug release at the cancer site. Also, this targeted therapy that nanomicelles can incorporate can also reduce other organ cytotoxicity. So that's why here we use nanomicelles. So in our nanomicelles, we used a mixture of two polymers. So this was a mixed nanomicelle. So a mixed nanomicelle can have a lower critical micellar concentration, and thus it can have a higher integrity as compared to a micelles made up from a single polymer. So here we use two polymers in our micelles. One was vitamin A TPGS, and the other was hyaluronic acid and PLGA copolymer. So vitamin E, these two polymers are amphiphilic in nature, meaning that they have both hydrophilic and amphiphilic groups. So the hydrophilic groups attach to each other, and the hydrophobic groups, they attach to each other. And the hydrophobic groups are in the core, and the hydrophilic groups surround the core, and they are present in the corona. That's why the nanomicellar structure, it solubilizes highly insoluble drugs like paclitaxel and ritonavir. And here in this also two drugs, that is paclitaxel and ritonavir, were added. And because these drugs are hydrophobic, they are concentrated in the core of the nanomicelles. And then these drugs are then solubilized by the nanomicellar structure. And when these polymers are present above the critical micellar concentration, they form nanomicelles. And here the red part of this polymer, that is vitamin E TPGS, it represents the hydrophobic part, while the blue part represents the hydrophilic part. And in the hyaluronic acid PLG polymer, the green part is the hydrophilic one, while the orange part is the hydrophobic. So as you can see, the hydrophilic part surrounds the corona and the hydrophobic part is in the core. And this is the structure for the nanomicelle. So first here we had to synthesize the PLGA hyaluronic acid copolymer. So for this, first we made the PLGA polymer by mixing lactite and glycolite in, and the reaction was catalyzed by stannous octet, and this formed the PLGA polymer. Then the PLGA polymer, specifically the end group was activated by EDC and NHS chemistry, and this made the PLG NHS molecule. And then this was then again reacted with, this was then kept aside. And then in a separate reaction mixture, we added hyaluronic acid. And then it was added with cysteine dihydramine chloride, again with EDC. So this gave us a combination of hyaluronic acid and cysteine. So here the cysteine bond was very important, because this is one of the important bonds that is cleaved in the cancer cells. Then this hyaluronic acid cysteine polymer and PLGA NHS, these two were combined, and this resulted in the formation of PLGA and NHS. Sorry, PLGA and hyaluronic acid. So this PLGN and hyaluronic acid, they were attached by a disulfide bond. And this disulfide bond is cleaved in the cancer cells. So cancer cells usually express glutamine synthase. And this is expressed 100 times more than the normal cells. So this bond is cleaved in, and so this bond gets cleaved in the cancer cells. And this helps to release the cancer drugs in the cancer cells and not the healthy cells. So if any compound has a disulfide double bond, we can say that it can reduce the cytotoxic effect that the cancer drugs usually have. Then we analyze the polymer hyaluronic acid PLGA using NMR spectrum. So here first, this is the red spectrum is for hyaluronic acid. And you can see that the peaks at 3.3 to 4 are the glycoside peaks for protons of hyaluronic acid. And then this long peak here at 1.8 is for the methyl group in hyaluronic acid. And the green spectrum here is for PLGA. And these two are the most significant peaks of PLGA, that is at 5.2 and 4.8, which is for the methane group of PLGA. And the purple peaks The purple spectrum is for the hyaluronic acid PLGA. And here we can see that we can see the spectacular peaks of PLGA, as well as we can see the peaks of hyaluronic acid. So this confirms that the copolymer, that is hyaluronic acid PLGA, was formed. Then we prepared the formulation. So this formulation was prepared by first weighing the polymers, that is vitamin E, TPGS, and PLGA and hyaluronic acid. And then these were dissolved in ethanol and a DCM mixture. And then the drugs, that is paclitaxel and retinovirs, were added. Then the solution was evaporated in a speed vacuum and under high vacuum and speed. Then this formed a thin film of the polymers with the drugs, that is the water insoluble drugs. Then this film was hydrated using the buffer, and then the buffer was passed through a syringe of 0.22 microns to maintain sterility and also to have nanomicelles of the same size distribution. So this method of nanomicelle preparation is called a solvent evaporation and film rehydration method. Then we wanted to optimize some of the formulation parameters. So for this, we use design of experiment by JMP software. So I have not shown the data here, but the entire data can be seen in the research paper that was published. So here we decided on some independent variables and then saw its effect on the dependent variables. So the independent variables were like, so the sonication time of the formulation and the polymer concentration. And the dependent variables or the critical quality attributes of the formulations were like size of the nanomicelles, polydispersity index, theta potential of the nanomicelles, and also the entrapment and loading efficiency for both of the drugs, that is paclitaxel and retinovir. So we played around with the independent variables, and then we got this form chart or the formula. And then we knew that, then I knew that when PLGA and vitamin E TPGS, if they are in approximately 1:2 ratio, the nanomicelles are in the size that we wanted, that is below 200 nanometers. So we saw that this particular combination of the formulation, that is 2 weight percent of HA-PLGA and 4 weight percent of TPGS, gave us the least size and the lowest PDI and near zero zeta potential, and it had the highest entrapment and loading efficiencies for both of the drugs, that is paclitaxel as well as retinovir. So we decided on this formulation, and all the rest of the studies were done using this optimized formulation. So this is how the nanomicelles look in a transmission electron microscope. So these black dots are the nanomicelles, and the size of the nanomicelles were 114 nanometers, which can be seen in this size distribution image. And they had a near zero zeta potential. And these nanomicelles then were incubated in a reductive environment. So this environment had a 50 millimolar concentration of glutamine disulfide. And so as I had mentioned earlier, so if the nanomicelles, they had this disulfide bond, these nanomicelles get easily reduced in an environment of glutamine sulfide. So we wanted to see if that theory was true. So in the first graph, so we have analyzed both the size of the nanomicelles as well as the amount of paclitaxel released. So in the first graph is the size of the nanomicelles. So the orange bars, the orange curve is for the final formulation, that is the hyaluronic acid, paclitaxel, and retinovir nanomicellar formulation in 50 molar of glutamine disulfide buffer. So when if we compare this to other formulations, for example, if there is no GHS, then the nanomicellar size remains almost the same. And also, if the formulation is without any hyaluronic acid is present in GHS, then there is no size difference as well. So similarly in here, we can see only when the formulation, that is the disulfide bond containing the hyaluronic acid, paclitaxel, nanomicellar formulation, only when it is present in glutamine disulfide 50 millimolar, we can see the highest amount of paclitaxel released. So this confirmed that our nanomicelle can only release the paclitaxel in cancerous cells. because they have high GHS. And here we wanted to see if the nanomicelles are effectively uptaken in cancer cells or not. So for this, we use 2 breast cancer cells, that is MCF7, which is a metastatic breast cancer cells line, and MDA-MB231, which is a triple negative breast cancer cell line. And we wanted to compare this with a normal breast cancer cell line, which is MCF12A. So for this, we first treated the nanomicelles with a fluorescent agent that was FITC. And then we saw time-dependent uptake of the nanomicelles in all of the cell lines. So in each of the cell lines, we saw the time-dependent uptake at 3 hours and 6 hours. And so we can see at, so as compared to 3 hours, in 6 hours, in all the cell lines, there was a higher uptake. And then also we compared the uptake for the targeted and the non-targeted formulation. So the targeted formulation at 3 hours was not much significant for MCF7, but at 6 hours, there was a higher uptake of the targeted formulation as compared to non-targeted. And then a similar trend was seen in all of the 3 cell lines. So this confirms that targeting with hyaluronic acid can have higher uptake of the nanomicelles in the breast cancer cells. Then we again wanted to confirm this uptake that was seen by fluorescence with a more sophisticated technique. And for this, we use uptake by LCMS. So here we developed a method for quantifying retinol and paclitaxel. And then we lysed the cells and extracted the paclitaxel and retinol drugs from it, and then actually saw how much of the drugs were there inside the cells. So just like the previous slide, there was a time-dependent increase. So first graph is the uptake for MCF7 cells. The B graph is for MD-MB231, that is a triple negative cells, and C is for the normal breast cancer cells. So in the the normal breast cells. So in the breast cancer cells, that is MCF7 and 231, we can see that with increase in time, there is an increase in uptake of the nanomicelles, targeted as well as non-targeted, as compared to the control. And additionally, the targeted nanomicelles uptake was higher in the breast cancer cells. And interestingly, we can see that in normal cells, at 6 hours, the uptake for both of the treatment, that is targeted as well as non-targeted, didn't have any significant difference. So that was good in a way, that the targeted nanomicelles are at least equal to the non-targeted ones in normal cells. So this can mean that the toxicity can be reduced for this treatment in the normal cells. So after this, we wanted to see if this therapy is potent enough to kill the breast cancer cells. So for this, we used 2 assays. And the first one was mitochondrial membrane potential, and the other one was reactive oxygen species determination. So if the mitochondrial membrane potential of a cell reduces, that means that the cell is going to die. So here we calculated the mitochondrial membrane potential of the breast cancer cell. So here again, we use 2 cell lines, that is MCF7 and triple negative breast cancer cells, and treated them both with various treatments. So in graph A, the difference in the treatments was the targeting. So we wanted to determine targeting with the mitochondrial membrane potential. So one of the treatment was plain nanomicelles, and the other was with the targeted nanomicelle, that is the hyaluronic acid. And then we can see in both the cell lines with hyaluronic acid targeting, there was a decrease in the mitochondrial membrane potential, meaning that this therapy was more potent for the targeted nanomicelles. And then here we had another variable, that was the retinol addition. Will this retinol addition enhance the effect of paclitaxel? So here we can see that the difference in these two is one of them has retinol, while the other one does not have retinol. And both of them, they have the hyaluronic acid targeting nanomicelles. So in the pink bar is for the treatment group that has retinol. So it was seen that addition of retinol decrease the mitochondrial membrane potential in both cancer cells, that is MCF7 and triple negative. And this can mean that additionally, in addition to hyaluronic acid targeting, Retinoid addition also decreases the mitochondrial membrane potential, which can be useful for killing cancer cells. Then in similar lines, we also saw the reactive oxygen species. If there is a higher amount of reactive oxygen species being generated, we can say that this cancer cell is about to die. So here in graph C, the difference in the treatment again was hyaluronic acid targeting. And then the pink bar shows increasing in ROS. And similarly for retinoid addition, there was also an increase in ROS. So we can say that here the increase in the active oxygen species can correspond to cell death. So here my project concludes, like these are the conclusions of my project. So the critical quality attributes of the nanomicellar formulation that was targeted with paclitaxel, and it had two drugs, that was targeted with hyaluronic acid, and it had two drugs, that is paclitaxel and retinoid. So this formulation had a mean size of 144 nanometers, had a low PDI, that is 0.2. The zeta potential was also near zero, which means that there won't be much aggregation of the nanomicelles. And also the morphology of the nanomicelles was spherical. So spherical morphology also helps in having a low aggregation. And there is, this helps in increasing the stability of the formulation. Also the entrapment and loading efficiency for both the drugs were like 95%. So that was also great. And also the in vitro uptake using LCMS and fluorescent microscopy, it depicted a time-dependent increase in the uptake of the breast cancer cells. And we also saw that the hyaluronic acid targeting as well as addition of retinoid, it helped in decreasing the mitochondrial membrane potential, as well as helped in increasing the ROS of the breast cancer cells, specifically the triple negative and the MCF7, that is metastatic breast cancer cells. So here we can conclude that the nanomicellar treatment that is both targeted as well as a potent treatment because of addition of paclitaxel and retinoid, it can be useful for metastatic breast cancer. And thank you so much. And I would like to take your questions. Thank you. Thanks. Recording. Aris, anu nahi kela kami?